Dr. Idit Goldfracht is a postdoctoral researcher at the Technion, at the Israel Institute of Technology. Idit completed her BSc studies cum laude in biotechnology and food engineering at the Technion, and her PhD research was conducted at Professor Leo Gebstein's lab in the Bruce Rappaport Faculty of Medicine at the Technion as well. Idit's research is focused on stem cells, tissue engineering, and heart electrophysiology. The results of her PhD studies won her several awards and led to high-impact publications in leading peer-reviewed journals such as Nature Communication. Idit is currently a postdoc in the Technion, where she is continuing her research in the field of tissue engineering. Her topic of presentation is development of engineered human heart tissue modeled from human stem cells derived cardiomyocytes. Idit, hopefully I said most of the names and of the things here correct. The floor is yours, please.
So hello, I'm Idit Goldfracht. I'm currently a postdoc in the Technion. I will present to you today the main finding of my PhD research, which was conducted in Professor Leo Gebstein and the Technion. The title of my talk is the development of an engineered human heart tissue model for human stem cell derived cardiomyocyte. In our lab, we're focusing on the use of stem cells. Stem cells possess two defining properties: the capacity for self-renewal and the ability to differentiate it into cell type with specialized cellular function. Pluripotent stem cell can turn into any cell in the body: bone, nerve, heart cell, and so on. In my research, I focus mainly on the use of IPS, induced pluripotent stem cells. IPS are stem cells that can be generated from somatic cells by a process developed by Shinya Yamanaka. Using this technology, it is possible to take skin cells, for example, from a patient and reprogram them into stem cells that can later be differentiated into numerous types of cells. In our lab, we are using this method in order to study inherited cardiac disorders. Since the patient disease is genetic, the stem cell that is generated from the patient sample will express it as well. Yet not every aspect of the human heart can be expressed and studied at the single cell level. Therefore, in my study, I combine the use of IPS with the tissue engineering technology. The general idea behind tissue engineering is combining a suitable scaffold with the right kind of cells in order to create the tissue you're interested in. In our case, that is a heart tissue. An engineered heart tissue can help in the study of inherited disorders, developmental biology, drug development, and regenerative medicine. There are numerous aspects and numerous concepts of tissue engineering, and our lab uses the hydrogel approach. Our model is the EHT, the engineered heart tissue. EHT was first published by Zimmerman as a collagen hydrogel-based engineered heart tissue in the shape of a ring composed of fat cardiomyocyte. In my research, I used IPS-derived cardiomyocyte in order to create this engineered tissue. My research goals were to generate and characterize a functional beating human engineered heart tissue, EHT, from iPS-derived cardiomyocyte. I then plan to evaluate the feasibility of using the EHT as a model for drug testing and for the study of cardiac disorders. In order to achieve these goals, the stem cells were directed to differentiate into cardiac cells. Here in this video, we can see a typical culture of cardiomyocytes that were differentiated from stem cells. On the right is a immunofluorescence staining of these cells for the cardiac marker troponin. We then use these cells together with an hydrogel, such as collagen, to create an engineered heart tissue, the EHT. The EHT was shaped as a ring and demonstrates continuous spontaneous contraction, as we can see in this movie. We then move to characterize our engineered tissue. We begin with immunofluorescence staining for the cardiac marker troponin and for collagen, and we compare the cells inside the engineered tissue to cell culture in a two-dimensional tissue dish under the same condition. We can see that the cells inside the engineered tissue are much more organized and elongated compared to cells in a culture dish. Further characterization was done using transmitted electron microscopy, TEM, and scanning electron microscopy, SEM. TEM analysis reveals the presence of cells in a much more mature state in EHTs that would culture for a longer period of time. SEM analysis shows the integration of the cells with the collagen fibers inside the engineered tissue. We then wanted to examine the electrophysiological properties of our tissue. To do that, we used a novel technology of genetically encoded calcium and voltage indicators. These indicators are composed from a sensing element that is fused to an autofluorescent protein that change its fluorescent level according to conformational changes in the sensing element. We can see here in the video an engineered tissue expressing the GCAM5, the calcium indicator. Every change in the fluorescent level is in correlation to changes in the calcium level inside the cell. Using a line scan in a confocal microscope, we were able to quantify these changes and to receive the calcium signal. We were then able to check the effect of different drugs on the beating rate of the tissue. For example, carbamylcholine, which decreases the beating rate, as we can see in the video and in the relevant signal, isoproterenol, which is similar to adrenaline and cause increase in the beating rate, and obain, which can cause irregularities in the calcium profile. We then wanted to check if our EHT model can be used as a platform for disease modeling. In order to do that, we use cardiomyocytes that were differentiated from stem cells derived from a patient with a cardiac disorder, long QT syndrome type 2. We use the arc light voltage indicator, and you can see here in this video a cell inside the EHT and how its fluorescence change just before the entire tissue is beating. The cells in the EHTs express the phenotype of the disease, meaning a prolongation of the action potential in a significant way in compared to a healthy control. We then wanted to take the model of engineered heart tissue one step forward and to address the different parts of the heart. The heart is composed of different types of cardiac cells. In most systems of differentiation, the resulting population of cells is a mixture of cardiac cells. This can affect drug development and disease modeling since they may not be accurate when mixed population is used. Our collaborator in Professor Gordon Carroll's lab in Toronto have developed a differentiation system which give rise to a pure population of either ventricular or atrial cardiomyocytes. We then use these cardiac cells, the chamber-specific cardiac cells, in order to create engineered heart tissue. Here in this video, we can see a ventricular engineered heart tissue, EHT, and atrial engineered heart tissue, and their action potential. The morphology of the action potential is similar to the relevant chamber of the heart. Using a fluorescent microscope, we were able to perform optical mapping of our tissue, which allow us to track the signal progress throughout the tissue. Here in the video, we can see an example of a ventricular electrical activity and its normal conduction. Atrial EHTs were mostly in arrhythmia, meaning a cardiac disorder caused by abnormal electrical activity of the heart. The arrhythmia was different from one tissue to another. In some, we saw multiple rotors, and in others, one single re-entry wave. Based on these videos, we were able to construct activation maps that allow us to track the signal throughout the tissue. In order to stop the arrhythmia, we gave it an electrical shock. Like in the hospital, we defibrillated it. Here in the video, we can see an example of atrial EHT in arrhythmia. We give it an electrical shock, and then normal conduction is resumed. We were then able to calculate the conduction velocity of our tissue and to compare the two types of tissue. We saw that for ventricular EHT, the conduction velocity is much higher than for the atrial EHT, and in both cases, it's increased with pacing cycle length. Finally, we wanted to study the mechanical properties of our tissue. The constructs were mounted between a force transducer and a post which position could be changed, thereby changing the initial length of the tissue. They were then subject to a series of increasing length while continuing measuring force. We saw a positive correlation between the initial length of the tissue and the force that it developed. This correlation is similar to the Frank-Starling relationship of the intact heart, and both tissues demonstrated it as expected. The ventricular EHT demonstrated a much higher active force compared to the atrial EHT, which is also in correlation to what is known of the native heart muscle. So to conclude, we established a functional engineered cardiac tissue model from iPS-derived cardiomyocytes. These models show potential for drug testing and disease modeling. We then wanted to examine chamber-specific EHT and use ventricular and atrial cardiomyocytes in order to do that. Atrial EHT were more predisposed to develop arrhythmia, and their conduction velocity was lower than ventricular EHT. Also, the tissue demonstrates a force-length relationship, which is a Frank-Starling relationship, when the ventricular EHT developed higher active force than the atrial EHT. We believe this engineered heart tissue model will help in drug development, disease modeling, and regenerative medicine. For further information, I invite you to read the articles that are published based on this finding in Nature Communication and in Acta Biomaterial. Thank you very much.